Hello everyone, my name is Prajakta Dhage and I'm from Department of Zoology, KTHM College in Nashik. Today I'll be taking a lecture for MSc Part 2, Semester 3. Subject is Soft Skills and this is the second chapter and topic that we are going to deal with is the stains and dyes. This is the 7th lecture of this particular chapter. Now let's move on to the dyes. Let's understand what are dyes. Many compounds are colored, but not all of them are dyes. A dye is a colored compound that can be bound by a substrate. In histological staining, dyes are used to impart color to various components of the tissues. Sometimes the coloring process has a high degree of chemical specificity, so that the dye can be used as a histochemical reagent. In many other histochemical techniques, the reagents are colorless, but the color substance are formed by reactions involving components of the tissue. Often these end products are insoluble compounds chemically related to dye stuff. Now let's talk about the general structure of the dyes. A dye molecule is traditionally described as having two parts. These are the chromogen, which is the colored part, and auxochrome, which is the part of the molecule that attaches to the substrate. The chromophore is the arrangement of atoms within the chromogen that is responsible for absorption of light in the visible part of the spectrum. The word auxochrome is used here as it is by dye chemist and histologist. Organic chemists use same word to mean an atom or a group of atoms that changes the wavelength at which a chromophore absorbs maximally. The auxochrome, which is a side chain attached to a chromogen, may be an ionizable group, or it may be a group that reacts to form a covalent bond with a substrate, or an arrangement of atoms that forms coordinate bonds with a metal or say mordant ion. Many dyes contain more than one auxochrome and most contain additional radical that modify the color of the complete compound. The color is also influenced by the number and types of auxochromic groups. The ionic charge of an auxochrome is balanced by an oppositely charged ion, most commonly hydrogen ion, sodium ion, or the chlorine ion. Dyes that have affinity to hydrophobic material generally lack ionized side chains. A fluorochrome absorbs ultraviolet, blue, or green light and emits light of longer wavelength. Fluorochromes that absorb in the ultraviolet are not or scarcely colored. These compounds are used in fluorescence microscopy. Some compounds become fluorescent only after they have been united with the components of tissues. Let's move on to another topic, color and chromophore. Compounds are colored because their molecules absorb quanta of electromagnetic radiation in the visible part of the spectrum. The energy absorbed by the molecules causes changes in the energy levels of electrons that are involved in covalent bonding and the lone pair of electrons associated with some atoms. With the organic compounds such as dyes, the absorption of visible light is largely attributed to the electrons associated with combinations of atoms known as chromophores. Chromophores are arrangements of the following atomic linkages. You can see the linkages over here. These linkages I'm talking about. These linkages I'm talking about. The double bond always alternates with a single bond to form what are known as conjugated systems. The bonding electrons are able to move from one atom to another along a conjugated system with the effect of exchanging the position of the atomic linkages formally designed as double and single bonds. This phenomenon is known as resonance. Benzene is a resonance hybrid of the extreme structures. Intermediate forms cannot be represented by simple structural formula. The equivalence of the bond in a ring such as that of benzene confers a stability that would not otherwise be expected in cyclic unsaturated compounds. Rings stabilized by resonance are said to display aromatic characters. The conjugated chromophoric system of a dye usually includes double bonds embodied in the formal structure of one or more aromatic rings. The resonance of the bonds in such rings commonly permits the existence of both aromatic and non-aromatic configurations in different rings within the same dye molecule. Let's move on to other groups, which is auxochromic groups. The auxochrome or colligator is traditionally held to be responsible for attaching the chromogen to the substrate. Other factors are also involved in binding of a dye, which we will be discussing later on. A basic auxochromes are amines, which can be protonated to form anions. A dye with a net positive charge is called basic or cationic dye. On treatment with strong alkali, the free bases, typically the unionized amines of such dyes, will be liberated. Acid solutions will favor the formation of cations. The colored ions of such dyes are usually balanced by chloride or sulfate ions. The acid auxochromes are derived from sulfonic or carboxylic acid or from phenolic hydroxyl groups. They occur in acid or anionic dyes, which may be encountered as the free acids or phenols or as the salts, usually of sodium. Sulfonic acids are strong, meaning that they are always ionized and exist as sulfonate ions even at the low pH. Carboxylic acids are weak, being only partly ionized in the aqueous media. Most phenols are acids, even weaker acids. Consequently, dye with carboxylic acid or phenolic auxochromes cannot exist as anion in the presence of high concentration of hydrogen ions. We'll talk about reactive auxochromes now. Reactive auxochromes are able to combine with hydroxyl and amino groups of the substrate to form the covalent bonds. Important reactive auxochromes include halogenated triazenyl groups such as dichlorotriazenyl, which is derived from cyanuric chloride. These auxochromes are usually attached to a nitrogen atom of chromogen. In the monochlorotriazenyl group, one of the chlorine atoms in the above formula is replaced by alkyl or an amino group. The chlorine atoms confer high chemical reactivity similar to that of acyl halides. Condensation with hydroxyl or amino group of the substrate with elimination of HCl occurs. Reactive oxochromes used in textile dyeing include pyrimidines and vinyl sulfones, which also react in alkaline condition. Dyes and other compounds with reactive oxochromes are not often used to stain tissues, but they are used extensively in the preparation of labeled macromolecules for use in nucleic acid hybridization and immunohistochemistry. The principal reactive oxochromic group for these applications are isothiocyanates, dichlorotriazenyls, and succinimide esters used because they combine with amino groups. Let's move on to another part, which is fluorescent compounds. The chemical feature that enables an organic compound to fluoresce are less easily defined than those responsible for color. In order to be fluorescent, a molecule must contain a system of conjugated double bonds in its hydrocarbon skeleton. The double bonds may include those of aromatic rings. Indeed, cyclic conjugated systems are associated with stronger fluorescence than are linear ones. Strongly fluorescent compounds, which are one of the interest in microscopy, usually have rigidly coplanar molecules. Thus, fluorine in which planarity is stabilized by methylene bridges is more strongly fluorescent than biphenyl, in which the two phenyl radicals are free to rotate about the single bond that joins them. Compounds with coplanar fused ring systems are more strongly fluorescent when the rings are all joined side to side than when there is a bend in the structure. Anthracene therefore fluoresces more brightly than phenanthrene in response to the same level of exciting illumination. That is, anthracene has the higher fluorescence efficiency of the two compounds. Fluorescence is also affected by substituents on the conjugated hydrocarbon skeleton of the molecule. When a single substituent is present, the fluorescence efficiency is usually increased. Alkali side chains generally have no effect unless they sterically hinder the assumption of planar configuration. The fluorescence of a compound can be influenced by other substances with which it is mixed and by the pH of the aqueous solvent or histological mounting medium. So this was all from my. Thank you so much for listening.